Hello everyone. In this recorded lecture, I will talk about the different techniques employed in the analysis of proteins. This is a continuation of our discussion about proteins. So we are already through with protein extraction. So now let us move on to the different techniques that we can use to further analyze our protein molecules. This recorded lecture contains 3 major protein analytical techniques. Now, there are other techniques available at present in protein analysis, but this lecture will only focus on SDS-PAGE, Western blot, and MALDI-TOF. So now let me first talk about SDS-PAGE. If agarose gel electrophoresis, or AGE, is the separation technique for nucleic acids, SDS-PAGE is the electrophoretic technique for the separation of the different protein fragments. Sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS-PAGE, is a high-resolution technique commonly used for separating the mixture of proteins according to their molecular weight. SDS-PAGE involves the use of polyacrylamide as gel and sodium dodecyl sulfate as ionic detergent to denature and linearize the proteins, hence named so. This technique was developed by Ulrich K. Lamley in the 1970s. When proteins are separated by electrophoresis through a gel matrix, smaller proteins migrate faster due to less resistance from the gel matrix. Other influences on the rate of migration through the gel matrix include the structure and charge of the proteins. However, for SDS-PAGE, the use of sodium dodecyl sulfate, or SDS, also known as sodium lauryl sulfate, and polyacrylamide gel largely eliminates the influence of the structure and charge, and proteins are separated solely based on the polypeptide chain length, or their masses. Sodium dodecyl sulfate, or SDS, is a detergent with a strong protein denaturing effect as it binds to the protein backbone at a constant molar ratio. In the presence of SDS and a reducing agent like 2-mercaptoethanol that cleaves disulfide bonds critical for proper folding, proteins unfold into linear chains with negative charge proportional to the polypeptide chain length. The binding of SDS to the protein molecules imparts a constant charge-to-mass ratio to the protein. Meanwhile, polyacrylamide provides a matrix for the polypeptides to run for their separation. Polypeptides run towards the positive electrode, or the anode, through the gel when electric field or electric current is applied. SDS-PAGE is performed using vertical electrophoresis setups, unlike the horizontal setups of the nucleic acids. In general, electrophoretic mobility is dependent upon the 3 factors, which include the shape, charge, and size of the molecule. For shape, it does not affect protein separation because after treatment with SDS and a reducing agent, all proteins are in the primary structure or in the linear structure. Similarly, charge also does not affect the separation of proteins because after SDS treatment, all proteins become negatively charged. However, for SDS-PAGE, size does affect protein separation. That is why protein fragments are separated solely based on their size or molecular weight after eliminating the influence of the other 2 factors. Smaller polypeptides move faster because they have to face less hindrance or resistance, while larger ones move slower because of greater resistance. Hence, proteins get separated only on the basis of their mass or their size. In performing SDS-PAGE, effective protein separation is contributed by a number of reagents. So the first reagent that I will discuss is 2-mercaptoethanol, or dithiotretol, or DTT. It is mixed in the sample to reduce or break down the disulfide bridges present between the polypeptides of a protein between cysteine residues. The disulfide bridges are responsible for the secondary structure of the proteins. Because electrophoretic mobility also depends upon the shape of the molecule, so it is very essential for all the protein molecules to be in the same shape. And this is achieved by using any reducing agent such as 2-mercaptoethanol. After the treatment with 2-mercaptoethanol, all the proteins should have a primary structure, hence the proteins are only separated on the basis of their size. Then of course we have the reagent sodium dodecyl sulfate or SDS. SDS is mixed in the sample buffer while preparing the sample for SDS-PAGE. After the treatment with 2-mercaptoethanol, proteins are in one shape, and after the treatment with SDS, all the proteins get a net negative charge. SDS imparts a negative charge to the protein molecules evenly. So when the voltage is applied, all the proteins migrate towards the positive electrode of the gel. And because proteins of different size get the negative charge proportional to their molecular weight, they only get separated by the size and not on the basis of the charge or shape. SDS is also present in the gel, making sure that all the proteins should stay negative throughout the gel. The next reagent that I will discuss is bromphenol blue or BPB. Bromphenol blue works as a tracking dye in electrophoresis and is used to monitor the progress of the molecules moving through the gel. It is mixed with the sample proteins, and when the voltage is applied, BPB or bromphenol blue runs along with the proteins but faster. It reaches the end of the gel before any protein in the sample does. Bromphenol blue has a slightly negative charge, that is why it moves towards the anode or the positive electrode, indicating the migration of the protein molecules. Another important component of SDS-PAGE is of course the polyacrylamide gel. Polyacrylamide gel is manufactured by the polymerization of the monomer acrylamide in water by using a small amount of a cross-linker such as NN'-methylene bisacrylamide. Hence, both acrylamide and bisacrylamide copolymerize and makes a 3D network of straight chain of acrylamide with interconnection of bisacrylamide. The ratio of the acrylamide and bisacrylamide determines the pore size of the gel. The method of protein separation developed by Langley utilizes discontinuous gels, which means that the method has both the stacking gel and the running or resolving gels. The migration of protein molecules vary greatly in the stacking and the running or resolving gels. These two gels have different compositions. When one wants to manipulate the gel pore sizes, adjustments on the acrylamide concentrations can be made. The purpose of the stacking gel is to stack the mixture of proteins that need to be separated on the interface of the stacking and resolving gel. Hence, all the proteins first get stacked on the line and then run into the separating gel approximately on the same time, no matter what the size of the protein is. The function of the stacking gel is based on glycine and chloride ions from tris-gly buffer and tris-hydrochloric acid buffer, respectively. When voltage is applied, glycine molecules and chloride ions begin to move through the gel towards the positive electrode. And since the glycine molecules are in the form of Zwitter ions in stacking gel, their electrophoretic mobility is very low. The chloride ions move faster than the glycine molecules. An area of unbalanced positive counter ions is developed, thereby developing a steep voltage gradient between the chloride ions and glycine molecules. Between the slow-moving glycine molecules and fast-moving chloride ions is the proteins from the mixture. Being intermediate in their mobility between chloride and glycine, the sample molecules or the sample proteins are carried along, becoming stacked into very thin, distinct layers in order of electrophoretic mobility. At this stage, The protein molecules are trapped between the two ion fronts, which is the trailing glycine and the leading chloride ions. Upon reaching the resolving gel, the pH increases and the pore size decreases abruptly. At much higher pH, glycine molecules are no more in the form of Zwitter ion, and they become ionized at this stage and start to move faster than their movement in the stacking gel. The chloride ions travel towards the electrode in no time as well. So, unstacking occurs and glycine molecules quickly overtake the sample protein molecules, leaving the protein molecules all free for the separation on the basis of their mass to charge ratio. In performing SDS-PAGE, there are 4 major steps. These are: first, the pouring of the resolving or running and stacking gels; second, loading the ladder and samples into the wells; third, running the gel by applying the voltage; and lastly, subsequent analysis by employing Kumasi blue staining. So for the first step of SDS-PAGE, the resolving gel is poured between 2 glass plates. When bubbles are formed, these are removed by adding a layer of isopropanol on the top of the gel. The gel is then allowed to solidify. When this happens, isopropanol is removed by using a filter paper. When the resolving gel is solidified, the stacking gel is loaded all the way to the top of the glass plates. The comb is then placed just after loading. The gel is then allowed to polymerize or solidify. When the stacking gel is solidified, the comb is removed very carefully so as not to damage the well's shape. So now let us move on to the second major step of SDS-PAGE, and that is the loading of the ladder and samples in the wells. So we add the ladder very carefully into the well, preferably on the extreme right, using a micropipette. The samples are also loaded into the other wells. The ladder is mostly pre-stained with a known molecular weight of proteins. The samples are loaded in each well with equal amount of the protein mixture, using also a micropipette. Take extra care in loading the samples, and make sure not to damage the wells or not to apply the sample out of the well for effective protein separation. So after applying the sample and the ladder into the wells, we then run the gel by applying the voltage. So a voltage is applied after dipping the sandwich of gel and the glass plates in running buffer. It is important to always turn off the voltage when the tracking die has reached or crossed the gel. This lets you know that your separation of proteins is almost complete. So the gel is further processed for the subsequent analysis, such as Kumasi blue staining. After running the gel, we then can proceed to the subsequent visualization of the separated protein fragments by Kumasi blue staining. In this procedure, the gel is rinsed with deionized water for about 3 to 5 times to completely remove SDS and the buffer. The gel is then dipped in Kumasi blue stain on a shaking incubator at room temperature. The invisible bands of the proteins begin to appear within minutes of the staining, but it takes approximately 1 hour for complete staining. So now let us move on to the second technique in protein analysis, which is Western blot. Western blot was introduced by Tobin and colleagues in 1979. Western blot is a commonly used method for protein analysis. It can be used for the qualitative and semi-quantitative protein analysis and post-translational modifications, such as in phosphorylation. In addition, it can also be used in medical diagnostics, such as HIV diagnosis. For the accomplishment of the Western blot, there are 3 elements involved. First is the separation of proteins by size. Second is transferring the proteins to a solid support medium. And lastly, the marking of the proteins by primary and secondary antibodies for visualization. There are 6 steps involved in Western blot, including sample preparation, gel electrophoresis, protein transfer, blocking, antibody incubation, and protein detection and visualization. So let's go over the different steps one by one. For sample preparation, proteins can be extracted from different samples such as tissues or cells. And since tissue samples display a higher degree of structure, the tissues are first broken down by the mechanical methods, such as the use of homogenizers or sonication. Protease and phosphatase inhibitors are commonly used to prevent the digestion of the sample at cold temperatures. After protein extraction, it is important to detect the concentration of proteins, which permits the determination of the exact mass or concentration of proteins to be loaded into each well. And usually a spectrophotometer is often used for protein concentration. So after sample preparation, we then proceed to gel electrophoresis, or the separation of the different protein fragments by SDS-PAGE. So the most commonly used gel is, of course, our polyacrylamide gel, and the buffers are loaded with sodium dodecyl sulfate, or SDS. Western blot uses 2 types of agarose gels. We have the stacking gel that is used to concentrate all proteins in one band, and the separating or the resolving gel that allows for the separation of the proteins according to their molecular weight. Smaller proteins migrate faster in SDS-PAGE when a voltage is applied. So polyacrylamide gel electrophoresis can actually separate proteins ranging from 5 to 2,000 kilodaltons according to the uniform pore size, which is controlled by the different concentrations of polyacrylamide gel. When we choose the appropriate percentage of the separating gel, we should always consider the size of the target proteins. The smaller the known weight of the protein is, the higher percentage of gels should be used. After separating the proteins by gel electrophoresis, the proteins are moved from within the gel onto a solid support membrane to make the proteins accessible to antibody detection. The main method for transferring the proteins is called electroblotting, which uses an electric field oriented perpendicular to the surface of the gel to pull the proteins out of the gel and move into the membrane. It can be done in semi-dry or wet conditions, while wet conditions are usually more reliable as it is less likely to dry out the gel. As shown on the figure, the membrane is placed between the gel surface and filter. The transfer sandwich is created as follows. So we have a layer of a fiber pad or the sponge, and then the filter papers, the gel, the membrane, and more filter papers, and the last layer of a fiber pad or the sponge. The next step is the blocking step. In this step, the membrane with the transferred proteins is blocked to prevent the nonspecific binding of antibodies to the membrane. Blocking only prevents nonspecific binding and does not reduce cross-reactivity. So what is cross-reactivity? It is the specific binding of antibodies to non-target proteins containing the same epitope as found on the target protein. So the most common blocking agents include protein-containing bovine serum albumin or BSA and the non-fat dry milk. The proteins from these blocking agents attach to all the places in the membrane where the target proteins have not attached. This produces clearer results as the background noise in the final product is reduced. After the blocking step, the primary and secondary antibodies are added. The primary antibody specifically binds to the target protein, and any unbound antibodies are removed by washing the membrane with a buffer. After the washing step, the secondary antibody conjugated with an enzyme is added based on the species of the primary antibody. Prior to the detection and visualization of the reaction in Western blot, a substrate is added for the enzyme to act upon. The substrate reacts with the enzyme that is bound to the antibody. Then, the color development is detected and should tell whether the protein target is present or not. Protein detection and visualization allows for the determination of not only the density of the protein targets, but also their location. So the size approximations of the targets are taken by comparing the protein bands to the marker. Western blot reactions are detected by using either of the following systems. So we can use colorimetric, chemiluminescence, radioactive, fluorescence, or electrochemiluminescence detection systems. Now let us proceed to another important technique in protein analysis, and that is MALDI-TOF mass spectrometry. Mass spectrometry is an analytical technique in which samples are ionized into charged molecules, and the ratio of their mass and charge can be measured. In MALDI-TOF, the ion source is matrix-assisted laser desorption or ionization, or MALDI, and the mass analyzer that is used is the time of flight, or the TOF analyzer. MALDI is a soft ionization that involves a laser striking a matrix of small molecules to make the analyte molecules into the gas phase without fragmenting or decomposing them. Some biomolecules are too large and can decompose when heated, and traditional techniques will fragment or destroy these macromolecules. So MALDI is appropriate to analyze biomolecules like peptides, lipids, saccharides, or other organic macromolecules. For the principle of MALDI, the analyte is embedded in a very large excess of a matrix compound that is deposited on a solid surface called the target, that is usually made of a conducting metal and having spots for several different samples to be applied. After a very brief laser pulse, the irradiated spot is rapidly heated and becomes vibrationally excited. The matrix molecules, energetically ablated from the surface of the sample, absorb the laser energy and carry the analyte molecules into the gas phase as well. During the ablation process, the analyte molecules are usually ionized by being protonated or deprotonated with the nearby matrix molecules. The most common MALDI ionization format is for analyte molecules to carry a single positive charge. It is believed that the first function of the matrix essentially is to dilute and isolate the analyte molecules from each other. This occurs during solvent evaporation and concomitant formation of a solid solution. Then, upon laser irradiation, it functions as a mediator for energy absorption. The choice of the right matrix is key to the success in MALDI. In general, highly polar analytes work better with highly polar matrices, and nonpolar analytes are preferably combined with nonpolar matrices. Currently, the most commonly used matrices are the alpha-cyano-4-hydroxycinnamic acid, 2,5-hydroxybenzoic acid, 3,5-dimethoxy-4-hydroxycinnamic acid, and 2,6-dihydroxyacetophenone. The time of flight analyzer is also a very important component of MALDI-TOF mass spectrometry. So the basic principle of TOF is that ions of different mass-to-charge ratio are dispersed in time during their flight along a field-free drift path of known length. Provided that all the ions start their journey at the same time, or at least within a sufficiently short time interval, the lighter ones will arrive earlier at the detector than the heavier ones. This slide shows the overview of the entire MALDI-TOF mass spectrometry process. So the analyte should be soluble to at least about 0.1 milligrams per ml in some solvent, and the matrix is dissolved to yield either a saturated solution or a concentration of about 10 milligrams per ml. The solution of the analyte is then admixed to that of the matrix, and then the mixture is then spotted onto a metal target plate for analysis. After drying, the mixture of the sample and matrix co-crystallizes and forms a solid deposit of sample embedded into the matrix. The plate is subsequently loaded into the MALDI-TOF instrument and analyzed by a software associated with the respective system. The MALDI leads to the sublimation and ionization of both the sample and the matrix. These generated ions are separated depending on their mass to size ratio through a time-of-flight analyzer, and a spectral representation of these ions is generated and analyzed by the mass spectrometer software, generating a mass spectrometer profile. These are the following applications of MALDI-TOF mass spectrometry. First is intact mass determination, and then we have peptide mass fingerprinting, oligonucleotide analysis, and lastly, MALDI imaging. The intact mass determination is basic and important for protein characterization because the correct molecular weight of a protein can indicate the intact structure. MALDI, a soft ionization technique, is suitable for proteins which tend to be fragile and fragment when ionized by other ionization methods. The performance of MALDI-TOF mass spectrometer is less affected by buffer components, detergents, and contaminants. In addition, it permits the intact protein mass determination with sufficient accuracy for the sequence validation. After protein digestion, MALDI-TOF mass spectrometry can also be used to analyze the obtained peptides for further primary sequence confirmation by peptide mass fingerprinting. MALDI-TOF mass spectrometry has simple operation, good mass accuracy, as well as high resolution and sensitivity. Therefore, it has widespread uses in proteomics to identify proteins from simple mixtures by a method called peptide mass fingerprinting, which are often used with two-dimensional gel electrophoresis. In this approach, peptides are generated by digesting proteins of interest with a sequence-specific enzyme like trypsin. And then peptides are analyzed by MALDI-TOF mass spectrometry to get the peptide masses. The experimental masses are compared against a database containing theoretical peptide masses from a given organism with the same sequence-specific protease. And with the development of molecular biology techniques and antisense nucleic acid drug technologies, more and more oligonucleotide fragments have been synthesized to be used as primers, probes, and antisense drugs. It is entirely necessary to quickly detect these fragments to determine whether the synthesis is complete and whether the synthesized sequence is correct. Mass spectrometry, including MALDI-TOF mass spectrometry, is by far the best means of doing this. Oligonucleotide analysis using MALDI-TOF mass spectrometry was simple, rapid, accurate, and sensitive, which can be used to determine the complete oligonucleotide sequence of our primers. The MALDI-TOF can also be used in profiling and imaging proteins directly from thin tissue sections, and this technique is known as MALDI imaging mass spectrometry or MALDI-IMS. It provides specific information about the local molecular composition, relative abundance, and spatial distribution of peptides and proteins in the analyzed section. MALDI-IMS can analyze multiple unknown compounds in biological tissue sections through a single measurement that can obtain molecule imaging of the tissue while maintaining the integrity of cells and molecules in the tissues. So this ends my discussion of the three techniques in protein analysis, which include SDS-PAGE, Western blot, and MALDI-TOF. Another important protein analytical technique is ELISA or enzyme-linked immunosorbent assay. This topic will be covered in your laboratory class. Thank you everyone for listening and enjoy the rest of your day.